On Zoom. I'm going to ask Scott to take a roll call and for Gil to conduct the meeting. Thank you.
Dan Bernal, Maria Bonneville,
Present.
Judy Chow,
Present.
Leandra Clark-Harvey,
Present.
Anne DeLiege,
Present.
Isabel Duron,
Here.
Mark Fisher-Colbury,
Here.
Fred Fisher,
Present.
Elena Flowers,
Present.
David Higgins,
Here.
Vito Bassiani,
Here.
Steve Julesgaard, Rich Lahara,
Present.
Chris Miaskowski,
Present.
Lauren Miller-Rogan,
Here.
Hugh Adriana Padilla,
Here.
Joe Panetta, Marv Southard,
Here.
Carol Watson, Kevin Zhu.
Good.
Okay, we have a quorum.
Good. Quorum is good. Thank you.
You did sound a little surprised.
No, I'm looking at this gallery view of smiling faces. All set. Is there anything preliminary before we start?
No, we can just turn it over to Gil.
Yeah.
To get a presentation.
We have a imposing agenda of 4 applications to review today. Gil?
Correct. Thank you. Good afternoon, everyone. So, today, Dr. Haley Lamb and myself are going to split those 4, and we're going to present each of us 2 applications to you. These are the recommendations from the grants working group that are coming from September and October reviews that we had. And let me just give you an overview to get us started. As always, we start with our mission, which is to accelerate world-class science to deliver transformative regenerative medicine treatments in an equitable manner to a diverse California and world. And with that, I want to remind you of the budget allocation that we have for the clinical program and show you where the status is as of today. So we had an allocation of $252 million for CLIN program. The amount that's requested today is $18.5 million. The amount of approved awards that we've done this fiscal year so far is 62.1. So if you are to approve the 4 applications, that would leave us with 171 million in our pool. The scientific scoring system that's used by the grants working group to score these applications is a system of 1, 2, or 3. Score of 1 means that the application has exceptional merit and warrants funding. There may be some minor recommendations that they make, but the grants working group does not feel it warrants them looking at it again. However, a score of 2 is one that needs improvement, and for those, we provide comments back to the applicant. Typically, they will revise their application and resubmit within a couple months. And then those that receive a score of 3 are deemed to be sufficiently flawed that they do not warrant funding at this time. The scientific review criteria that informs that score is based on these 5 questions. Does the project hold the necessary significance and potential for impact, meaning what value does it offer, and is this something that's worth doing? Does it have a sound rationale? Is it well planned and designed? And is it feasible, including having an appropriate team assembled to conduct the work, as well as having appropriate resources to do the work? And then lastly, does the project uphold the principles of diversity, equity, and inclusion, or DEI? In addition to the DEI that's considered by the scientific reviewers, we also have a separate DEI score that is given by our patient advocate members of the board. The DEI score system is 0 to 10, with 10 being the best possible score. And there's a rubric that we have created to help inform how the scoring for the DEI is done. So you will see 2 scores for each application, one which is the overall scientific score, and then a separate DEI score. The composition of the working group includes scientific members who give that scientific score. They have varied expertise that we bring to the table in order to assess these applications. In addition to them, to those 15 members, we have 7 board members who are patient advocate or nurse members of the ICOC. They do the DEI evaluation, provide patient perspective on the significance and potential for impact, and also oversight on the process. And then on occasion, we have scientific specialists who are non-voting members. They provide a scientific evaluation in areas where we have gaps of knowledge or where we need to bring in additional knowledge related to any particular area. And for those members, they do not provide a final scientific score. Okay, so we're going to take each application one at a time. And the first one has the following conflicts, so just keep this in mind. We're going to show you before each application who may have a conflict with that application, so please refrain from discussion or making comments related to this application as we discuss it. So this is CLIN 1 14945. It's entitled Clinical to Clinical Gene Therapy Development for CMT4J. This is in adenovirus 9 gene therapy. This is for patients that have CMT4J, which is a mutation in what's called the FIG4 gene. I'll tell you a little bit more about it in the next slide. The goal of this study is to complete IND enabling activities and to file an IND. The funds requested are just under 4 million, and they provide co-funding of 982,000, which is the 20% that's required for these applicants. All right, so a little background on this disease. It's Charcot-Marie Tooth Disease Type 4J is the formal name, abbreviated CMT4J. And this is an ultra rare disease. It is debilitating, progressive, hereditary motor and sensory neurological disease, and it's caused by a heteroallelic FIG4 gene loss. And this is characterized by progressive motor weakness with some sensory involvement resulting in quadriplegia, respiratory failure, and a shortened lifespan. And currently there are no treatments available to slow or halt the disease progression. It is simply treating the symptoms as best as one can. The value proposition for this therapy, this proposed gene therapy approach utilizes AAV9 vector to target affected neurons and provide that missing FIG4 gene. The approach has the potential to significantly improve the quality of life for those patients who are affected by this disease and possibly halt the progression of the disease. Why is this a stem cell or gene therapy? Well, this is a gene therapy approach that uses AAV9, so therefore qualifies for term funding. This particular disease is indeed ultra rare, and so it is not currently represented in any of our active TRAN or CLIN awards. And this particular applicant has not previously received a CIRM award. The recommendation from the grants working group indicates that they voted unanimously for a score of 1, with 15 members giving it a score of 1, and the DEI median score was a 9. And the CIRM team recommendation is to fund this application for the award amount of $3.93 million. And so I turn it back to you, Mr. Chairman.
Great, let's start with comment. Can I be heard?
Yep.
Yeah.
Yes.
Start with comments from the board members, please.
I have one question.
And Marie, please.
Yeah. Can you just tell us what is the estimated prevalence of this disease in California? Roughly how many patients may have this particular mutation?
Yeah, it's a great question. It's hard to know in California specifically. So the prevalence is about 1 in a million. So it's an ultra rare indication. There's just over 30 patients that are part of a group that are known to the applicants that they will tap into to gather those that would participate in the future trial that they're preparing for. And the future trial would enroll about 20 patients ultimately if they can manage. They are also doing a natural history study, which includes 20 patients that is part of this group. So the number of patients in California, I don't have the information, but of patients known, it is so rare that I don't know whether there are any or how many there would be in California.
Yeah. No, I really appreciate this comment, Gil. And the reason for my question is clearly based on the scoring from the GWG. It's something that we would want to fund, the fund request. My only question is, will the team be able to enroll 20 patients in the actual trial plus 20 patients in the natural history study, given how ultra rare that is? Is it even feasible?
Yeah, that's also a great question. And they have already identified the patients for the natural history study. So they have those already. And so I think that was part of the confidence that it gave to reviewers of them being able to enroll the 20 that they need for the actual clinical study once that comes into place.
Okay, so without spending too much time on that, I just wonder if that should be taken into account in our reasoning to support the grant or not. And Gil, you're so much experience or others, which is I have no question about the science and the judgment of the GWG. Should we fund a trial where we know that they're going to struggle enormously to enroll this study, and patients easily participate in a natural history study? It's another story to enroll in a gene therapy, even if their hope for a better life is desperate. So I welcome your opinion, Gil, or anyone else for that matter. How realistic is the trial? Is my question.
May I say something?
Yeah, I'm sorry. Yeah, I know, of course. Sorry, I didn't realize you were here, actually. Sorry about that.
Yeah, I thought I was on the Zoom. Anyway, I'm sorry, Anne-Marie. Actually, the investigator already lined up 2-3 patients to come to California, or one of them is in California. And so they're bringing the patients to California for treatment.
Okay. Okay, so you're confident that they can bring a total of 20 patients. Okay.
Well, no, they're going to treat, as Gil described, they will do a natural history study, but they're going to treat up to a certain number, which is not all 20. Remember, they need to identify with the natural progression of the disease as similar to a placebo control, and then also treat up to maybe maximum 6 or a little bit more with this gene therapy as a comparator. So that's the way it will work.
Great. Thank you for the clarification.
You're welcome.
Any other board members have comment?
David Higgins. David Higgins and Judy Chow.
David Higgins is next.
Judy
Thank you. Not to assume that these researchers don't know what they're doing more so than even we do. Can you go back to them and say, because this is so rare, you need to ask for more money, or we need to give you more money in the upfront? And if that is it true that more money equals more patients, or is that a pipe dream? So my question is, should we as the funding agency tell them what they need when we think they don't understand what they need?
So, David, I think that's a good question, but I think, part of the assessment of these proposals include what their ultimate plan for enrollment and recruitment of these patients would be. And so they address part of that in here. Just remembering that this is a Clin 1. So this is for the IND enabling work. This is not yet for a clinical trial. So they would not do the clinical trial until they complete these initial studies. But part of what they did share in the application is their partnership with patient advocacy organizations. With patient advocacy organizations. It looks like I'm echoing a lot here. All right, I'll try it again. Thank you. And so they have patient advocacy organizations that they have partnered with in order to help identify future patients. So there is a plan in place. So they do have an idea of what to do, but they don't have to do it at this particular stage.
Michelle?
Judy?
I have a follow-up question. So then if I understand from Apple's response, the study will be done in California, or this is going to be a global study? And then even if at this stage is going to be state administrated study, realistically moving forward, do they have plan like then this is going to be a global study to increase the number of patients to be able to demonstrate the efficacy?
Right. So the company is a California organization. So they have clinical site in California and one outside of California that they would plan to use, and probably will increase the number of sites beyond that. So it would be, in terms of the patient population that they would recruit, would be global, but there's going to be a limited number of sites that include California.
I see.
Yeah, and Judy, if I can say, the actual applicant already lined up all those patients. It's sort of like he has been the patient advocate on behalf of all the families that have those types of patients. So essentially the patients have been identified, and it's almost like a case study on its own. The actual champion of that effort, who set up the company called Alpida in the state of California, is who applied on behalf of all those families.
So more or less the budget, including to be able to fly the patient to the studies, all already included?
No, because this is not for the clinical trial. This is for IND enabling work. So the clinical trial is not something that would be funded under this award.
That'll be the next step.
That correct.
Good. Thank you.
I don't see any other hands. Do we have public comment?
We don't have a motion.
We don't have a motion.
Yeah, motion, yeah.
I move to approve funding for this. Clin
Southern
1.
seconds.
Fred Fisher and
Mark Southern.
Further discussion from board members on the movement to accept 14,945. Okay, if not, public comment? Does anyone see any?
No.
You don't?
No, there are no hands raised.
Okay, Scott, I think we can proceed then.
Judy Chow,
Aye.
Leandra Clark-Harvey, Anne-Marie DeLiege,
Yes.
Mark Fisher-Colbury,
Yes.
Fred Fisher,
Yes.
Elena Flowers,
Yes.
David Higgins,
Yes.
Vito Imbasciani,
Yes.
Rich Lahara,
Yes.
Chris Miaskowski,
Yes.
Lauren Miller-Rogan,
Yes.
Adriana Padilla,
Yes.
Joe Panetta,
Yes.
Marv Southard, Carol Watson. Are there any board members whose names I have not called who are not in conflict? Great, thanks very much. The motion carries.
Okay, carries. Good. Thank you. Gil, you may proceed.
Okay, thank you. So these are the conflicts for the next application. And the next application is CLIN 15060. The title is a 1XX enhanced and fully non-viral BCMA chimeric antigen receptor or CAR-T cell therapy for relapsed and refractory multiple myeloma. Therapy is a CAR-T, cryopreserved autologous track locus BCMA CAR-T cells. The indication is, of course, for relapsed and refractory multiple myeloma. And the goal is here, once again, to complete IND enabling studies and to file an IND for a future clinical trial. The funds requested are 4.585 million. There is no co-funding required for this applicant. Little background, multiple myeloma is the second most common malignancy among the blood cancers and primarily affects individuals that are over the age of 60, the median survival being about 7 to 10 years. And there are several treatments that are available, but patients are typically will relapse and become refractory to additional lines of therapy. And on average, a patient will go through 4 to 5 different lines of therapy during the course of multiple myeloma. The value proposition for this proposed therapy, CAR-T therapies that target BCMA have demonstrated significant promise for treating relapsed and refractory multiple myeloma, but the current approaches still have limitations. One of those is in particularly the relapse that often occurs. And the proposed therapy aims to overcome these limitations and produce a CAR-T therapy that would be more persistent and reduce relapse. It also offers the possibility of making the manufacturing of the therapy more efficient and less expensive. And so why is this a stem cell or gene therapy? This is a therapy that's composed of T memory stem cells. It is also a therapy that would be considered a gene therapy because of the genetic manipulation in creating the CAR-T. This particular, well, we currently don't have any active TRAN or CLIN projects in multiple myeloma at this time, but we have had some very early stage and in the discovery stage, and have had some in the past that were later stage. This applicant has not previously received a CIRM award. And here's the recommendation from the grants working group. The grants working group members voted with 11 members giving this a score of 1. There were 3 that gave it a score of 2, and the DEI was a score of 9. The CIRM team recommendation is to fund in the amount of 4.585 million. And back to you, Mr. Chairman.
Thank you, Gil. Having heard the presentation on 15060, the chair will entertain a motion.
Southard moves approval.
Marvin has moved.
Second.
David Higgins.
Yep.
That was David, okay. So motion's moved and seconded. We can start with discussion from board members.
I don't see any hands at the moment.
I don't see any hands. I'll give it 5 seconds. Okay. Can you check for public comment at this point? No hands.
No hands.
We're not seeing anything. All right. Then I think we can proceed, Scott, with our vote.
The motion is to fund CLIN 15060.
Correct.
Judy Chow,
Yes.
Leandra Clark Harvey,
Yes.
Anne Marie Deliege,
Yes.
Isabel Duron,
Yes.
Mark Fisher Colbury,
Yes.
Fred Fisher,
Yes.
David Higgins,
Yes.
Vito Imbasciani,
Yes.
Steve Julesgaard. Steve, I don't know if you're on mute.
Yes.
Great, thank you. Rich Lahara,
Yes.
Lauren Miller Rogan,
Yes.
Adriana Padilla,
Yes.
Joe Panetta,
Yes.
Marv Souther,
Yes.
Carol Watson.
The motion carries.
Motion carries. Very nice. Gil, would you take us into the third application presentation?
Yes. And so Dr. Haley Lamb is going to present these last two. Haley?
Good afternoon, everyone.
Haley?
So the third application is CLIN 15085. This is a personalized antisense oligonucleotide therapy for rare pediatric genetic disease, SCN2A. And so the title is, I think, somewhat self-explanatory. This is a therapy that's a personalized antisense oligonucleotide drug, and the indication is SCN2A associated genetic disorder. The goal of the project is to complete a first-in-human trial, and they're requesting just under $1 million in funding with no co-funding required and none provided. A little bit of background on this. So the disorders are named as such because there are mutations essentially in the SCN2A gene. And so the class of disorders results in kind of a range of neurodevelopmental conditions and mainly are characterized by how severe the epilepsy is. That's a side effect of it. So the severe forms of this disease cause seizures beginning in infancy, and commonly the typical anti-seizure medications are not effective. So for the proposed therapy is an ASO that would treat a single patient with severe epilepsy and severe neurodevelopmental delay. And if it's successful, other people with similar disorders could potentially benefit from equivalent precision therapies. And this is a gene therapy product. CIRMS does not currently fund any active TRAN or CLIN awards for this disorder. And the applicant has received prior CIRM funding with an active discovery stage award. This is candidate discovery earlier stage, and it's about halfway through right now. And finally, the grants working group recommendation was a tier 1 to recommend for funding and a DEI score of 8.5. And the CIRM team concurs with the grants working group for the award amount of just under $1 million. Back to the chair.
Thank you. Would like to have a motion to consider this application.
Isabella's raising her hand.
Isabella's her hand. Well,
Isabel, you wanted to speak?
I was going to ask a question, but do we wait for the motion first?
You could do both.
I could, all right. Well, let me ask the question first then. Given 6 were a 2, can you describe, either Gil or Haley, what the hesitation was?
Yeah.
Seems high, but 6 still, wasn't top of the line. So what were the hesitations?
Yeah, so the main concerns of the panel were around the monitoring of the subject after treatment. So the clinicians wanted there to be EEG monitoring of the seizures post-treatment because they felt it would be more accurate. The current protocol asks for sort of parent-reported seizures, which the panel thought may or may not be accurate, because parents are not necessarily with the person all the time and maybe can misidentify, that sort of thing. So they wanted the applicant to revise the EEG monitoring from, I think they had one time point at 12 months, so they wanted a couple of additional time points added.
I don't recall the DEI score. Can you remind me what that was?
Yes, I believe it was 8.5.
And so we know that this is a problem that exists across racial ethnic groups.
I think it's, I would say, so according to the applicant, they've only had 9 cases identified with this genetic disorder. So I'm not sure if there's enough data on that.
Considered rare. Well,
Yes.
I'm wondering how they got an 8.5. If the researchers themselves can't identify that there's a racial ethnic issue involved here, or this is specific to one group or a specific group of children. How do you get an 8.5 if they don't know?
Well, I think, Isabel, I think that the problem is that it's so rare with less than 10 patients worldwide that it would be hard to know whether it impacts a diverse population or not, right? So just based on the 10 patients, they can, give you numbers there, or they can give you a distribution of what may exist there, but it's not going to be predictive of what may exist worldwide.
Mm-hmm, mm-hmm. But nobody asked about the different patients or the breakdown of just those 10 to know, that there was some diverse. I know you talk about rarity, but I'm curious how you get such a high score when you don't have the data to substantiate or not that they in fact have a DEI plan, that they understand. Sorry. I'm confusing myself.
So yeah, I mean, I think I would if you look at the comments, I think from the DEI sections that the applicant institution has a strong DEI track record, and they have language support for the various possible patients that they could potentially draw upon. And yeah, so it was, I think, with the caveat that I think the panel understood that this was very rare. And there was, one comment that, the identification of the disease means that there has to be genetic testing. And so that does have a cost associated with it.
Right. But that shouldn't leave people out because they can't afford the cost. I mean, I'm saying.
Right.
I'm a shade confused here. Okay, proceed.
Let me ask again for a motion to consider.
You have 2 hands up. 3 hands up.
Yeah, I have one.
I think there are more questions than there are. We're ready for a motion just yet.
Okay.
Yeah. Thank you, Steve.
Yeah.
Application slide. Thanks.
Okay, Emery, you have a question.
Yeah, I assume that we should vote on whether we approve the additional funding for the study, and we should not question the original idea of the study. Is that right?
Gil?
Is that correct? Yeah.
I'm sorry, could you repeat that? I didn't understand.
I just want to clarify what we're voting on, and I'm pretty sure. But I assume we're voting for allowing additional funds to the tune of less than $1 million to be given to finish the trial. We're not voting nor even questioning the original funding of the trial. Is that right? It's a supplemental funding.
No, this is a first in human trial that's going to treat 1 patient.
Mm-hmm.
I see the N equal 1.
Correct.
Oh, thank you. So it's the original trial, and the cost is close to $1 million for this particular patient.
Correct.
Yes.
So then my question becomes more relevant and valid, which is, I don't know of any intervention that can tackle a development delay when it has already happened. At best, you can prevent progression of development delay. I don't think you can ever reverse one, but maybe I'm wrong. So is there a value? And it's not the amount of money, because of course, relatively speaking, compared to the rest of the grants we're making, this is rather small, but is there value in spending money of something I don't believe can be achieved? It would be a totally different situation if the indication was severe, intractable seizures with a goal of preventing development delay. But I don't think that's what I've heard. And I have a question about trying to achieve something that, to my knowledge, and I'd love to be told again that I may not know it so well, cannot be achieved.
Right. So that there was some discussion amongst the panel on this issue, and it's basically unknown. They don't expect there to be recovery of neurodevelopmental delay that's already happened, but they do believe that it's possible that the epileptic episodes could be reduced, and so therefore could have benefit in that sense. That was a comment from one of the.
I wonder if others and you and Gil can comment your own opinion about that, because there are things we know and things we don't know. In fact, there are lots of things we don't know. I believe that if you stop a process such as intractable seizure, you can stop the progression of development delay. I've never heard, really at all, of a situation where you can reverse a development delay. And particularly in children, I don't know the age, and I don't know also the seriousness of that particular person development delay. I just have a little problem of voting money for something that is undoable. However, the endpoint of intractable seizure is a more doable endpoint.
Steve, your hand is up.
Yes. So I want to actually, this dovetails a little bit with Isabel's question, but it really is a different point. This is a study, just to be very clear, this is a study in only one person with an extremely rare disease. There are, if you read the whole review that the GWG provided, there have been only 9 people identified in the whole world with this condition.
Mm-hmm.
So extremely, extremely rare. So now this is new ground as far as I'm concerned for us.
Yes.
So we're getting into the business of providing potential treatment for one person for a disease that hardly ever occurs. And the question gets to be, if this works, are we now bound to provide support for this person for the rest of their life? I'm going to assume that they're not going to be able to find very many other people around to be treated. If we're talking on a global basis, only 9 or 10 people like this. And this is something we should be doing. I don't know that I have an answer to that, but it's, we're getting ourselves really into the questions of these very, very rare diseases. What role are we going to be playing? And I understand this is a really difficult disease, particularly for the family of this patient. But on the other hand, we're now potentially lining ourselves up to become the payers. And if CIRM, winds up ultimately not getting funded again in the future, the parents are going to be in a position of trying to figure out how to pay for this therapy, because I don't know that insurance will ever pay for it. In any event, I'm a little bit troubled by this, and I think it's worth thinking about a little bit, just the nature of this disease and the rarity of it, and what things look like down the road.
So can I just speak to that briefly? I can't answer the question about whether it should be funded or not for this particular award, but this is for the trial of the single patient, and there is a foundation that's partnering with the applicant that has at least in a letter of support, has committed to provide the treatment to the patient for their lifetime.
Great, that's helpful. I didn't see that anywhere.
Thank you, Steve, for those important comments, and for Haley. I'm going to go to Maria and then Fred Fisher.
I had similar comments that Steve had. I also, just from a philosophical perspective, is CIRM going to continue to fund applications that come in that treat a single patient? And that calls into question whether, how quickly we can get a strategy around a rare disease, how we fund rare disease, where it's going, and how the benefits can be shown to help in other disease indications, because oftentimes that is the case. So it puts us into, it sets a precedent for us. If we fund this, it's meritorious. What do we do in the future? And I'm not suggesting we shouldn't fund it. It's just a philosophical question, and I think that's something that the board and along with the internal team needs to have a discussion around at some point.
Fred?
Yeah, so this is a very interesting discussion, but I would suggest this is not the place for the discussion that's happening. We've spent an inordinate amount of time talking about something that is not the purview of this committee. Although I agree that Steve and Maria have proposed substantive topics that the board might choose to chew on at a board meeting where this issue is agendized. Because where the line is drawn between rare disease, ultra rare, extremely rare, N of 1, these are distinctions that are not necessarily clear in everyone's mind, and certainly are not grounded in policy. So let's take this conversation to the policy place, which is the board, I think. And let's focus on this application, which got 8 of the scientists saying, this is a great idea, let's do it. And 6 saying, this is a really good idea, but needs a little tweaking here and there in their minds to get it fundable. And my guess is the CIRM staff are completely capable of addressing the issues that were identified by the 6 in order to satisfy anyone who has concerns that the vote was divided 8-6. I would be saying something completely different if it was divided 8-6, with the 6 being 3 and the 8 being 1. So we don't even have a motion on the table, which I think should actually precede these kinds of discussions rather than come after the fact. But that's, again, another thing that's up to the chair to decide. But let's just focus on this application and consider it the merits of this application. Thank you.
Leandra, before I get to you, let me make a comment. If first of all, Fred, I tried to get a motion. I agree with you. I think this is a discussion of the motion. Number 2, I like your suggestion that this should be this should be chewed over. I think those are your words, by the entire board. So I think I will ask the science subcommittee to bring this issue to the board for a full discussion that's duly agendized. Thank you for that. Leandra?
I was just going to support that as well. I do feel like I feel uncomfortable voting because I know what our mission is, right? But this is really a question around how do we enact that in this case? And I do feel that we need to be really clear and level set with a greater board, as Fred has suggested, and others, around what that looks like for us, because this may come up in the future and I'm not comfortable setting a precedent in this meeting.
Thank you. I'm going to ask our board counsel here a question now. If no motion comes to accept this, does this go into a limbo for future discussion? Maybe after the board has a deeper conversation on this topic?
Well, you could table consideration of this until such time as the larger conversation happens. if there is no motion. Typically, we would, if there is no motion to fund, we would entertain a motion not to fund, but it's up to you and how the.
Yeah, I like that suggestion. Maybe the board members heard that. If someone would like to table a decision on this application to a date certain, no, to a date uncertain, but not to be forgotten.
I'm happy to entertain that motion that this a decision on this particular application should be tabled until the board had a chance to review the entire philosophy around ultra-rare disease.
Thank you, Emery. We have a motion?
Second, Fred Fisher.
Seconded by Fred. Okay, does that, a little rusty on Robert's rules, a motion to table, does that have to be voted on?
So you can ask if there is any objection to tabling the matter.
Thank you. You heard board count. Is there any objection to our tabling this issue? Oh, thank you so much. Okay, good. We have tabled it. Gil, I'm going to ask you then to go on to the fourth and last application.
It's Haley.
Hmm?
Haley's.
Oh, did I skip?
So there's one conflict on this application, and the application is CLIN 215115, the CURE trial, cellular therapy for in utero repair of myelin. I can't say it. I'm sorry.
MMC.
It's essentially spina bifida. And so the therapy is allogeneic mesenchymal stem cells on a matrix graft. And the goal is to complete a phase 2A clinical trial. The funds requested are just under 9 million, and the applicant has provided just under 6 million of the co-funding, which is 40% required. So a little bit of background. So this is a birth defect that occurs with an incomplete closure of the spinal cord while it's developing. And it results in damage to that exposed spinal cord, and the damage leads to lifelong lower body paralysis and bladder and bowel dysfunction if it's not treated. So the current standard of care is in utero surgery, which improves the quality of life of children born with this condition, but over half still cannot walk independently. So the value proposition of the proposed therapy is that it could improve the motor outcomes of the children born with this condition compared to surgery alone. And ultimately, if successful, would result in more children who are able to walk. And this project is composed partly of the mesenchymal stem cells together with the matrix material. Similar CIRM portfolio projects, there's essentially the phase 1 part of this current application under your consideration that is finishing up, and this is for the same product for the same indication. And the prior CIRM funding to the applicant team, essentially this particular project has been funded by CIRM since the preclinical stage and has progressed to what you're considering today. And the grants working group is a unanimous recommendation for funding of a tier 1, a DEI score of 9, and a CIRM team recommendation to fund as well for, again, the total amount of just under 9 million.
That's it, Haley. Thank you. Right? Great. Okay.
That's it for me. Thank you.
We have a motion to consider discussion of this final application.
So moved.
Maria Bonneville has moved. Do I hear a second?
Second.
Elena, thank you. Right here in the room. Board members. Comments on this in utero surgery for spina bifida? No. Okay. Any members of the public want to chime in? Do you see anything? No. Nothing? Okay, nothing. Scott, ready for the vote?
The motion is to fund CLIN 2 15115. Maria Bonneville,
Yes.
Judy Chow,
Yes.
Yvonne McCarty,
Yes.
Anne Marie Galeage,
Yes.
Mark Fisher Colbury,
Yes.
Fred Fisher,
Yes.
Elena Flowers,
Yes.
David Higgins,
Yes.
Vito Ambassiani,
Yes.
Steve Julesgaard,
Yes.
Rich Lahara,
Yes.
Chris Miaskowski,
Yes.
Lauren Miller Rogan,
Yes.
Adriana Padilla,
Yes.
Joe Panetta,
Yes.
Mark Southern.
Yes.
Great, thanks very much. And the motion carries.
Great. Gil or Haley, any final remarks?
No, thank you very much for your time and your discussion. Thank you.
Okay, discussing the work for the chair is to assign the topic of the treatment of N-1 applications to come to the board via the science subcommittee. With that, I know we're going to have one more board meeting in December, but this committee will not meet again until the new year. So if I don't see any of you on this subcommittee before 2024, happy holidays to all.